I'd like to thank the organizers for giving me the opportunity to talk about the work that I've been doing within the Bioenergy Science Center towards metabolic engineering of Clostridium thermocellum for biofuel production from cellulosic substrates. Just briefly, the Bioenergy Science Center is a DOE-funded, multi-institutional center with the aim of understanding why plants are difficult to break down and then engineering around it using biotechnology. And this collaboration involves over 300 individuals working at over 17 institutions. And our overarching theme is to make better plants, better microbes, and then to interface the two using better tools and better combinations of microbes and plants to really enable biofuel production from plant material. I work within the microbial focus area of BESC, specifically engineering Clostridium thermocellum for enhanced yield. Clostridium thermocellum is one of our primary model systems, and there are a number of reasons for that. It's a thermophilic anaerobic member of the firmicutes, and it can be isolated from a variety of different environments, including compost piles or hot springs. And it's a model system for cellulose breakdown because it contains this extracellular protein complex that coordinates the activities of numerous biomass solubilizing enzymes. And so you get a synergistic effect between different types of cellulases such that they can better break down plant biomass and solubilize it. And then C. thermocellum can then ferment those sugars into fuels, for instance, ethanol. But one of the big challenges is that ethanol is only one of many products that it makes, and so it will require metabolic engineering in order to turn it into a strain that can take cellulosic biomass and actually convert it at high yield into a fuel. C. thermocellum has a mostly Embden-Meyerhof type glycolytic pathway, although I'll talk about some of the idiosyncrasies later at the very end. In addition to making ethanol, it also produces acetate, lactate, formate, and hydrogen as primary fermentation products. But more than that, it also produces a variety of apparently overflow metabolites, where there's some sort of metabolic bottleneck, and the cell responds by just dumping things out into the supernatant. And one of the main things that it produces as part of this overflow metabolism is amino acids. It secretes amino acids similar to Corynebacterium glutamicum. And this can account for, in a wild-type strain, up to about 10% of the carbon flux. So our goal is to eliminate all these other products and streamline metabolism such that we make high levels of ethanol and then eventually other end products as well. So building a platform that we can use for design of pathways for more advanced fuels and then also bulk chemicals and so forth. Our collaborators at Mascoma Corporation did a lot of the fundamental work on building genetic tools to be able to manipulate this organism. And their initial proof of principle using these tools was to knock out the carbon pathways for the combination of lactate and acetate. And the idea was, well, if you knock out these primary fermentation products, then you should just get a lot of ethanol. And this has happened successfully in other organisms, including ones that Mascoma has worked on, like Thermoanaerobacterium. Unfortunately, knocking out these two pathways didn't really increase ethanol production all that much. The wild type and the genetic background that's used, a delta HPT strain, they make ethanol, which is green here. The double knockout, it makes a little bit more, but not a ton more. And we don't really know why, but the strain also grows really poorly. They evolved the strain to grow better, and when they did, the strain made substantially more ethanol, but it's still only at 50% of theoretical yield. And again, we don't really know why that is, but it seemed to kind of max out at that point. And so I started working on this project as a postdoctoral researcher in Lee Lin's group, and we decided to take an approach of, instead of trying to constrain carbon flux, let's constrain electron flux, because conversion of glucose to ethanol is perfectly redox balanced. And so if you're making other reduced products like hydrogen or formate, then when those electrons get lost to the environment, the cell is no longer able to make ethanol using those electrons, obviously. And so if we can limit the flux of electrons to things like hydrogen and formate, we should be able to increase ethanol production. And so we started by knocking out hydrogenases and the pyruvate formate lyase. And then we continued engineering this strain to also lack the lactate production pathway and the acetate production pathway. And when we grow these strains on the first side is cellobiose, a model soluble substrate, we see that metabolism shifts dramatically from a mixed acid fermentation to primarily ethanol as a primary fermentation product. But this is still only about 50% theoretical yield, similar to what Mascomo saw in the evolved strain. And this also holds true on model cellulose substrates. This is sigma cell here, where we see a little over half of the carbon going to ethanol production. This also holds true for pretreated substrates. So this is the cellobiose and the sigma cell crystalline cellulose. And then in white is acid pretreated populus, and then in black is acid pretreated switchgrass. And while the wild type converts some of it to ethanol, the engineered strain produces more than twice as much from each of the pretreated real-world substrates. Similar to the Mascomo strain, this, we're calling it the quadruple mutant, it grows a lot more slowly than the wild type. And this is presumably due to a metabolic bottleneck of some sort that's preventing flux from the sugars into ethanol. And we reasoned that if we can evolve the strain to grow faster, that the strain must be debottlenecking itself. And so then we can investigate what mutations accumulated in the evolved strains to infer what the metabolic bottlenecks are in the original mutant, and then use that information for further metabolic engineering. And so what we did is we took the original mutant, inoculated 2 tubes, and then we just transferred those 2 tubes in separate lineages pretty much every day. For 300 generations, that's 30 transfers, all the way up to about 1,500 generations, around 150 days of transferring the strain every day. And the strain, you can see in light blue is the original mutant. It doesn't even really have a true exponential phase, but even after just 30 transfers, you get a good logarithmic growth, and the growth rate is substantially improved. And then after 150 transfers, 1,500 generations, the growth rate is even faster, and you get a good growth curve. The second lineage here in orange, it did improve, both at 30 generations and at 30 transfers and 150 transfers. But interestingly, lineage number 1 did substantially better. And if we look at ethanol production from these evolved strains, here's the wild type, here's the original unevolved mutant. This is ethanol concentration on the y-axis and number of transfers on the x-axis. You can see that after just 30 transfers, there's about a 30% increase in ethanol yield that corresponds with this increase in growth rate. Interestingly, lineage number 1 continues to get better as we improve the strain, whereas lineage number 2, for some reason, starts doing worse as far as overall ethanol yield. So we're actually in the process right now of resequencing at numerous points along this evolution, and we hope to be able to correlate those mutations with the changes in phenotype. Okay, so I said that C. thermocellum has a mostly Embden-Meyerhof type glycolytic pathway, but there are quite a few idiosyncrasies, and some are potentially rather subtle, like using GTP or pyrophosphate instead of ATP as a phosphoryl donor. But then there are also other more major differences, like the lack of a pyruvate kinase. Most organisms have a pyruvate kinase, our bug doesn't. And instead, it probably goes through oxaloacetate and potentially malate, which could cause a variety of different redox imbalances that I don't have time to get into. And so this is where we're really starting to team up with JGI and Sam Deutsch in particular and the gene synthesis team to start exploring the metabolic diversity that's out there in thermophiles, both in cultured thermophiles and in metagenomic libraries, to start synthesizing potential solutions to what we imagine are metabolic bottlenecks here. And so we've started receiving some constructs that we've designed from JGI. And just as a baseline, this is that quadruple mutant with an empty vector in it, and it's still making just under 30 millimolar ethanol. But 3 of our clones in particular, each of which represents a different strategy for fixing metabolism, each of the 3 shows approximately a 25% increase in ethanol yield. And so we're in the process of characterizing these strains and the effect that they have on central metabolism, and then also trying to stack these mutations into a single strain to see if there's a synergistic effect. And so we look forward to working with JGI a lot more for gene synthesis to try to fix a lot of the problems that we have with C. thermocellum. We're also using the gene synthesis for trying to improve genetic tools and a variety of other things that we want to fix about C. thermocellum. And so with that, I'll acknowledge the people that did a lot of this work. Well, first I'd like to acknowledge my postdoctoral advisor, Lee Lind. He continues to be a collaborator, even though I'm now at Oak Ridge National Lab. Ranjita Biswas is the postdoc that made most of the mutant strains that I talked about. And Nanan Jiang is a postmaster's student who did the bulk of the strain evolution part. And then again, thanks to Sam for all of his help.
Have questions for Adam? I guess I have one. This central metabolic pathway is pretty well trodden ground over the last century. Rather than using an evolutionary strategy, is there a role for metabolomics to identify where some of these bottlenecks are, or what intermediates might be accumulating that might allow you to save some time just going in to knock them out directly?
Yeah, so that's an approach that we're also taking, although metabolomics has a variety of technical challenges associated with it. Extracting metabolites from E. coli or yeast or other model organisms is pretty well established at this point. But one major area that Lee's group has actually been looking at is how to quench metabolism rapidly and get the metabolites out in C. thermocellum, because it's a lot more difficult of a bug to crack than E. coli, for instance. But that said, we are using metabolomics to try to investigate where these bottlenecks are.